So I heard from Matt that he will not be joining us today, but Dimitri is supposed to join. I see Dimitri is there. So I think Dimitri will convene today and Erasmus MC will take minutes. Dimitri?
Een heel goede reden om eens te beginnen.
We'll just wait till Dimitri is back. Dimitri? Dimitri, can you hear me? Dimitri, can you hear me?
Having connections problems, trying to get the camera on. Hi. Hello, everyone.
Hi.
Sorry about that. Hi.
Matt told me that he will not be in and that you would convene today. Is that correct?
Yes. I just thought we had a meeting at the end of the month, so sorry about that. Okay. But I'm okay with moderating the meeting today. No problem.
Okay, I will take a minute then.
1 second. I'll tune in and tune out and tune in. Just 1 second. Good. Yeah. Thank you for waiting. So hello, everyone. We'll probably start straight away. Welcome to the monthly meeting. How many participants we have today? Can everyone hear me? Sorry, I had connection problems, so I just need to make sure. Okay, great, good. So I'll just share the screen just to see what's on the agenda today. Okay, so let's start then. Probably on series 1, the SAR gaps of the compounds and actions from the last meeting. Does anyone have any updates on this? Hank?
All right, so I've ticked that off. Firstly, hi everyone. I think I've seen a few new faces that I might not have met, but yeah, my name is Hung and I'm from UCIT. I am a PhD student. My supervisor is Alice, who's actually joining today, and I'm also working with Clay myself at University of Sydney. So if I haven't seen you before, I just want to say hi and good day. So yes, I've ticked off the first few ones. So the first one is the SAR diagram. So from last meeting, what was missing was if you scroll down to the bottom, Dimitri, I should have already have a update to post. Yes, that one. So basically what was missing was the thing, the activity. So we just basically need the mind map will help us work out like which compounds within the epichem libraries will we should be asking and which like what kind of priority. So there were 17 compounds that I and Dimitri have compiled together. From there, I've narrowed it down to 11. So the 11 that are that I've highlighted in red, and I've also written a post down the bottom as well why that we should be asking for them. So we're trying for 10, but I think 11 might work a little bit better. So it's not down. So if you scroll down, I've already updated the comment down the bottom as well. So those are the one we suggest, Dimitri and I suggest that we should be moving forward for asking, was it Martin or someone at the Epichem library to get that for us? So I've done that one. So that's the first line, the first action item on the agenda.
So do you know who should be asking for those compounds? Is it
I
going to be?
believe it would be test to Matt, but gotta double check with Matt when he's available.
Okay, so I'll talk to him when he's back from that meeting about this, because the same applies to the manuscript. Just a short update, like on our side. So the manuscript has been is awaiting evaluation, just a final review by Matt. So there will be no amendments at this point until he has like a final look, and then if something pops up, then we will amend it on the go. But should be pretty much done, I guess. So experimental, the second bullet point about the experimental section, this has been done before the previous meeting, so that there shouldn't be any issues with that as well. All the data and significant figures. There are a few comments left, but I'm pretty sure that's fine, because we discussed this with Matt as well. The offline copy of that notebook, so I can see that Hank, you uploaded like some time ago. I'm pretty sure I did mine as well, but I double check because I don't remember, to be honest, but I'm pretty sure because I did update all the lab archives files.
Yeah, so for me, I've already put my what's relevant to the manuscript number 2 onto the USIT repository, and that's the link for that. So I've also put that into the manuscript itself as well. So that should be fine from me. So yeah, just double check, Dimitri, if you have already.
And I'll provide the link as well for the under this issue as well. Yeah. The next point about the biological supplementary data, Wendy, I need to ask you on this because I'm slightly confused. That's the first time I'm seeing this. Can you elaborate on which spreadsheet we're talking about? Because I got slightly confused on this.
Yes. So for us, we have to make an offline copy of the spreadsheet. So we will copy the data we have in Excel. Then after Matt runs through the whole manuscript, we go over it one more time to see if all the molecules which are included are also present in the Excel sheet. And then we will finalize that Excel sheet, and that will be the supplemental data for the biological screening. I'm sorry.
So essentially, all the molecules that go from UCL and from University of Sydney should be in the spreadsheet that we should provide for you to double check. But all the SMILE strings, all the data essentially from the MycetomaTis spreadsheet.
Yes, so we make sure that everything is there, and but the molecules which might be sent afterwards, which were not included in this manuscript, we will take out, of course. So we will have to take out, we will only include all the Fennery molds.
Is it supposed to be online version, like a new page on the Excel file on the MycetomaTis spreadsheet for us to share?
Yes, so we could refer, of course, to the current spreadsheet, but most manuscripts also want an offline copy which you can download so that it's fixed, and that's why we would make a copy which is uploaded at that time, but it's fixed, so you cannot alter anything anymore.
So probably on GitHub, sorry, on Dropbox, we should upload a file on Dropbox then, okay?
Yeah.
Sure. The review of the final version of the manuscript, we talked about this just a few minutes ago, so this will be done in due course as well. Any updates on the in vivo data from Erasmus MC? On the larva season, on the potency, I think. ISOMA published a PowerPoint presentation earlier today on this with some.
Yeah, so Ma will give her updates soon. But the larvae season will start this month. So we ordered the larvae, and the first experiments will be done next week or the week thereafter.
Sure. So going further on the new transcriptomic data, is there anyone specifically who was assigned on this on bullet point number 2 about the
Well,
grain?
I made the agenda. The transcriptomic data is also generated by us, so I think that will be me. So we did perform a transcriptomic analysis in the Galeria melanella model. And there we looked at different time points. Sorry, I'm just going to ask if they could be a little bit more quiet. We did do a time course event where we infected Galeria melanella with Madurella mycetomatis, did isolate RNA of host and pathogen, and analyzed what kind of transcription we found. And one of the things we found in the larvae was actually that the fungus produces siderophores. So we found a lot of transcripts of the siderophores. And the interesting thing of siderophores is that they could enter the grain probably. And that's why we thought it might be cool to look if the siderophore could act as a Trojan horse, so that you could use it to couple an antifungal agent to and get it inside the grain. And that's one of the downsides we currently have with the Fennery moles. So some go in, others don't go in, while they have very potent in vitro activity. So I thought if we could couple them to a siderophore, maybe they can get in the grain and we can get activity again. Because the siderophore will be taken up by the fungal cell itself, internalized, and then the antibiotic or antifungal agent can do its activity. And that was a hypothesis I had, and I thought maybe this could help there. And I saw that Bernard already responded to this on GitHub. So maybe Bernard, you would like to respond here as well?
Yes, as you have mentioned, I've already added some links to your issue about that matter. And yes, I might ask some of our colleagues from the former network, because we worked on that project, or colleagues of mine worked on this project, on such a project by coupling siderophores to penicillins, for example. So it should be quite possible to conjugate Fennery moles to such hydroxamide-based siderophores, for example. And maybe because two of these colleagues are from the University of York, and we already have a contact at York about the Series 2 project, maybe that might be most promising to ask these two colleagues first about that matter.
I have a quick question for Bernard. Technically, how difficult would it be, and time-wise, in terms of like how time-consuming it would be to make those molecules in the lab?
Of course, this was a PhD project, but now it's published and it just needs to synthesize or to use these published protocols, which are quite straightforward, and it's not too difficult to prepare.
There is one.
I think best way is to connect it via the Fennery moles, via the piperacin ring systems, where there are some carbamate residues, and to replace the carbamates by a linker and the siderophore.
Yeah, and the fungal specific siderophore you can actually buy from Cayman chemicals.
Yeah, it depends if they can be modified then accurately. But
Well,
of
I think
course,
they
that's
obtained
a
it from that
possibility.
other manuscript where they looked at antimalarial agents also from there, and then they modified it.
Okay, yeah, that's a good point, yeah, to start with.
So does it mean ideally we take the molecules already from the existing library, say from the ones that I synthesized at UCL or at once at the University of Sydney, and then we do like some sort of like coupling, and we test them in biological assays? That's how it would work.
Yeah, so in the ideal situation, we take those Fennery moles which were very active in vitro, but didn't do anything in vivo, and then coupled those Fennery moles, and of course a negative and a positive control, so a Fennery mole which didn't do anything at all in vitro, and one which was potent in vivo and in vitro. You would couple them then, and then we would test first if they still have their in vitro activity. And if that's the case, then we would test them in the larvae and see if we then get enhanced larval survival. If that's the case, that would mean that the Fennery mole, which was potent in vitro and could penetrate the fungal cell in vitro, can now also penetrate the grain, so can pass through the cement material and enter the fungal cell. Because there's no reason to think that the Fennery mole itself would not have activity, because we already demonstrated that it had activity in vitro, but it couldn't penetrate into the fungal grain. So that would be an ideal group of molecules to look at. Of course, we have other molecules of interest which could work as well. But since we took so many time looking at the Fennery moles and we found this log D correlation, the next question would be, how can we circumvent that, and can we couple them towards a carrier or a Trojan horse, in this case the siderophore, and can enter the grain then, because that would in the future help with any other molecule we will find, because it's not so difficult to find molecules which have in vitro activity. It's more difficult to get molecules which can actually penetrate the fungal
Penetrate the
grain.
grain. Yeah, that's the problem I've been having all the time.
And if this would help, that would open a very large subset of molecules we could actually use to treat fungal infections, or mycetoma in this case.
And the reason why
So
I
I
asked
thought,
Bernard about applicability in terms of chemistry is because I still have some time left in the lab, but I just want to be realistic. Like if we go for this option, it sounds very interesting and I would love to do it, but I'm not familiar like with how long would it take. But my understanding is like we already have a number of molecules synthesized like in the lab, so we need to do like some modifications and we should be ready for the assay. So maybe that would be even better for the project rather than doing like new analogs in the months that are left. But I'll have a talk with Matt and we'll consider this option as well. It sounds very
I think it's a very cool option, and the Fennery moles are very excellent candidates for this because we did so much work on them already.
The only problem which we might have is the fact that the molecules are not stable. So the ones that have been synthesized, like for example, 2-3 years ago, we would have to check again whether they're still haven't degraded. So that's the only possible issue. Otherwise, it should be fine. Okay, I'll put this on discussion list with Matt for sure.
Dimitri and Bernard, it's a nice suggestion. I think, Dimitri, that's great for you to go ahead and speak to Matt. It makes great sense for you to take that on if it fits with the time frame, and if Bernard and colleagues at York are happy with that approach. Alternatively, or complementarily, we could maybe do a little bit of that here at Sydney, perhaps with some of Hung's compound. So do just let us know.
Sure.
I'll
keep you updated on this.
Yeah, sure. No pressure, but we might have some capacity for that too. So would be good to know what you decide with Matt.
Yeah, just on
Great,
that. On that note as well, I do have some compounds here over at Sydney as well that I know are still from a couple of years ago that are still quite I checked them, so it still look good, which means that like if the siderophores route is going to be, something interesting, then I'll probably won't do it because I'm already at the end of my PhD, but like someone else can pick that up and I can just hand those over to whoever is interested in continuing that work, so I'd use it. So yes, it sounds like a great idea.
But it might be also other series, molecules from other series used as well, the ones that have been proven like potent, like aminothiazoles potentially, right? Like ketoxams as well. So anything. Okay, yeah. Very interesting.
It doesn't matter which series it is actually, but I just opted the Fennery moles because we have so many molecules. But if it's another series because it works chemically easier, that's also fine. But I just like the idea of using the cytophore as a Trojan horse.
Yeah, we can have a look at that, Wendy. We've got certainly some compounds from series 2, from the SSP students, and Marita and Payan are working on some other series too. So we could follow up on that and see if there's a potential for us to work on that. It's a nice idea.
All the compounds from UCL are still in the lab because I did like a larger scale synthesis. So they should be, most of them, available as well, I would propose. Okay, moving on to the next part then. So update from the university. Sorry, series 2, meeting between chemistry team and high schools. Alice, I'm not sure, were you involved in this or was it Kimberly? And
I unfortunately wasn't able to attend, Dimitri. Kimberly did. She's on holiday at the moment. So I think we might have to wait for a little update there. Clementine is here.
Yeah, I can.
I think you were at that meeting, weren't you, Clementine?
Yes, I was at that meeting. I mean, we basically just discussed all of the different targets that the various schools and us at UCED are going to be aiming for in the next few months. And I haven't actually updated the page yet, but I'm about to update that page. I just need to get the right permissions on the GitHub. And so everything that is being made by various different schools will be on there for people to look at.
okay. Update from the University of Sydney. Okay, we just had this about the high school. Any updates on series 5? Moving on to the next part on potencies. But I think, Wendy, you said that we need a bit more time on this, right? For you and Ma to update us on the potency results. Or do you have any new data to share?
Well, maybe it's good now that Ma might share her presentation so that she can talk about all the things we've done for the in vitro stuff. So, Dimitri, could you unshare so that we can share?
Yes. Yeah.
So, can you see our screen?
Yes.
Yeah, thank you. Yeah, today I want to show my compounds data from MycetOS Open Source compounds. Firstly, have a small review, small summary. Our compounds include series 1, series 2, series 5, and series 6. This one just came from last summer. Yeah, and 7 natural products. And the MMV Global Healthy Priority Books. It includes 300 compounds. At first, those compounds from series 1, and those are from last 2 years. Then we updated the MIC50 of those compounds. All data are were updated in the spreadsheet.
So, for those who are new, so the MIC means that we did minimal inhibitory concentrations to different isolates. The IC50 is that we look only at one isolate and look at which concentration 50% of the cells is inhibited. Here we look at total inhibition of each of the isolates. And you see that there are different between isolates. So I11 is a little bit more resistant than I1, for instance, for this compound. And then we take the median, and that's the MIC50. So that's the concentration in which 50% of all isolates are inhibited by that certain compound.
Yeah, thanks for Wendy's introduction. Yeah, then yeah, those compounds worked with lower MIC50. We tested in vivo test last summer, but yeah, the in vivo data were also in the spreadsheet. So as for the series-1 compounds, we get series-1 compounds this 2023. Then 10 compounds we selected to do the MIC50. We can see those all worked very well to inhibit the Madurella mycetomatis isolates.
Yeah, and they range from 0.125 if you look at this compound, so that's very potent, and this one is a little bit less potent.
Then it's a compound from series 2. We both tested the MIC test against the MM and FS. Yeah,
MM is Madurella mycetomatis.
Yeah, Madurella mycetomatis. Yeah. As for series 2, we tested them against two strains. Madurella mycetomatis is MM. The phosphorus nigrolensis is FS. Yeah. And then as for the MIC test for Madurella mycetomatis, we select 5 compounds. And their MIC50 was lower. Just like this one, MIC50 is 1 micromolar. These two a little higher, but those are below 8 micromolar. Yeah, and then we tested those against the phosphorus nigrolensis. We select 3 compounds inhibited fungi growth at 25 micromolar. Then we do the further test to get the IC50. Then this is those three compounds were inhibited. These 3 compounds inhibited fungi growth at lower concentration. So these 3 compounds from series 2 will be tested in vivo this summer.
So just to summarize these 3 compounds, so 421, 425, and 426 were inhibiting both Madurella mycetomatis and falciformis spora senegalensis, and the MIC was roughly the same in both species, so they were very potent.
Yeah. Then is a compound from series 5. We get series 5 compounds this summer and last year. As for this year, this one, the MycetoS 328 and this one 333 and MycetoS 334 looks very promising. Yeah, very promising to test in vivo. But yeah, and then we tested it against the FS to narrow the number to do in vivo test. Those compounds from series 5 were tested by Mimi, last summer. Then I also tested the MIC50 for those compounds. then just 1, 2 compounds looks promising. As for those compounds, they were tested against phosphomysporous negligence. Then those compounds from last summer, there are no compounds inhibit fungi growth at 25 micromolar, but those 3 compounds inhibit fungi growth at 25 micromolar. Then those 3 compounds were tested at from 16 to 0.03 micromolar. Then they both inhibited at 8. Then these 3 compounds were also inhibited Madurella mycetomatis growth. So these 3 compounds, a little summary. These 3 compounds inhibited fungi growth at lower concentration against Madurella mycetomatis and phosphomysporous negligence. The next one is series 6. Just 1 compound left from last summer. Yeah, the MIC 50 data we need to update. So it will test this summer. Then it's about the nature products. As for 4, 5, 6, 4, 5, 7, 4, 5, 8, 4, 5, 9. Yeah, those are active against the MM and with a lower MIC 50 against Madurella mycetomatis. Those are were tested against the FS, and I didn't. I forgot to show the data. And these 4 compounds are still inhibited fungi growth at a lower concentration against Madurella mycetomatis. A little summary is in vitro test. We did MIC 50s for MM recently, and all data were updated in GitHub. As for the series 2 and series 5 compounds, 3 compounds were selected from IC50. Then there were test. They will be tested in vivo. As for the MMV Global Health Priority Books, the MIC data at 125 micromolar for Madurella mycetomatis and phosphomysporous negligence were updated in GitHub, and 7 compounds were 7 compounds inhibited both strengths at 25 micromolar. So those were selected to test the IC50s against both 2 isolate strands. Then the experiments are ongoing. In vivo test plan, and the 3 compounds from series 5 will be tested in vivo larvae model against Madurella mycetomatis infected larvae and phosphomysporous negligence infected larvae. And same with series 2 compounds. And for nature products will be done. Will be tested. Will be tested in this larvae model. Also. Okay. And maybe we will get several compounds from the MMV Global Health Priority Books. So let me see. That's all in my slides. Thank you.
Thank you, Ma. Wendy?
Yes, so this was an overview. And of course, there are many compounds still to be tested in vivo, but we will start with that upcoming weeks.
Wendy, just a quick question. What about the compounds that we sent from UCL back in March? Have they been tested in vitro yet? What's their status? On the Caterpillar Series 6.
No, sorry, I have no time to test them yet.
So no, they are still on the to-do list.
Okay, I've seen that Mr. Tariq Khan has joined the meeting. Is he going to test some of the new test compounds which were not tested yet in the coming summer, or what's the current state?
For this summer, it was difficult to find a time where both Tarek and I will be together in the lab here. So probably that will be a little bit later. But we will try to do our best to get everything tested as soon as possible. But we want to make use of the larvae season as well, of course. Tarek, would you like to please go ahead?
I'm sorry for not being very much connected to your group earlier and not having a very in-deep idea how the things are moving in this MycetOS group. Currently, as long as my introduction is concerned, I'm Tariq Ayub. I'm working in a university in Saudi Arabia. I'm a biochemist basically, and our previous work is on parasites. We do in vitro activities on a parasite like Leishmania and Toxoplasma. However, I'm now connected to this group, and I'm very much interested to take on the studies on mycetoma. Currently, we're looking for the cases in Saudi Arabia, as this is not a very common disease, and the climate of Saudi Arabia itself is not really favoring for this disease to take its root. So at the moment, I'm just exploring. I'm connecting different hospitals, their databases. I'm connecting it to different research centers in Saudi Arabia, whoever has ever come across this case, so that we can take on, and at least we can get some isolates from here, and we culture them here and start working on them in Saudi Arabia as well. And definitely we'll be collaborating with the laboratory elsewhere to get their the help and the guidance how we can carry on.
I have a question because ATCC, the American Cell Culture Organization, has 2 strains, mycetoma strains from Saudi Arabia in stock, or does it offer? Have you any update on this? Could you get a strain from them?
Yes, they do have. They have 2 strains. One strain is available for the shipment. However, they have to fulfill some shipment requirements, and they have to get like 2 extra bit of permissions. I have contacted them, so they told me that they have to get 2 extra permissions. So this is one of the option. But before we go for that, I'm looking for existence of some active cases in Saudi Arabia, so that at least it could get some attention from research centers here that this case is existing. Previously, there are some studies. The most recent study is in 2015, and they have identified 19 cases of mycetoma. Of them are 7 fungal, and this study was done in Jeddah. But after that, there is no study talking about mycetoma at all. And that was like more epidemiological study to see the occurrence of mycetoma in Saudi Arabia. But currently, I'm looking for some active cases so that I could get the isolates out, and I could identify what kind of the causative agents are there causing this mycetoma in Saudi Arabia.
Yeah, the other option of getting strains is the Fungal Biodiversity Center in Utrecht, which also has a large collection. That could be another option, of course. But I think it's good to search for active cases as well, because then at least it would be a very fresh isolate and not a laboratory strain.
Yeah. Because this strain, which Dr. Bernhard, indicated, it was done almost 30 years before. 1987, there were like some cases, in some hospital in Eastern Province. So it was isolated at that time. I'm not sure whether, even if we get those isolated, whether we'll be able to culture them here or not. Not sure about that. So better if we go for some active cases. As I mentioned, that it will also, gain some attention from the authorities, so we can, get some support from them as well.
Okay, thank you. Then we'll probably move on to the next part of the meeting, and that's so we discussed series 5 a bit in the presentation. A question about oxazoles. So Bernard and Andrea are on this issue. It says here on GitHub that they would check with the NDI. Bernard, do you have any updates on that?
Sorry, no, they told me that they will do the new compounds this spring. So maybe in early summer they are ready for shipment. But I will ask them again. Don't know if any one of them is here in the meeting. Don't think so. But of course, I can write an email and ask for them for these compounds again. What's the current state of synthesis and so on?
They have to be synthesized. It's not from the library, right?
And Louisa's here, so maybe she can answer.
Oh, okay.
Yeah, I mean, I don't, well, for those who I don't know yet, I'm Louise. I'm replacing Ben Perry from the NDI, from the MycetOS and the OSN. So I don't know, I didn't know, I wasn't following up this, so probably Andrea is the best person to ask about this oxazoles. Regarding we have something to share, if we could send compounds. I think that honestly, what I can say is that it's very difficult to get compounds out of the NDI inventory to go to groups. So what we can do first is try to set up an MTA, material transfer agreement between us and probably Erasmus University would be the best option. But I've been trying to do this for one of compounds. I forgot the idea of the compound from that we published in the JMedChem paper last year, and I'll be trying to do this with the legal department for almost like 6 months now, and it's very difficult to set up an MTA, even though the compound has been published and everything. So I wouldn't go that route, maybe relying on us to share compounds, because it's very difficult and they're not very, it's not that simple, just send compounds. I wish it was, but it is not. And what we can do is, for example, I know that Wendy has asked me if we had some compounds, some approved drugs in our inventory. For example, we do not have that, but what we could do is try to help with the costs of buying those. We have some budget to do reimbursement for compound, like buying compounds for like Enamine or any like commercial library. So we could do that for sure. But sending compounds, if you give me like a structure and I can look for in our inventory for sure, if it's something like that you guys really want to, I can look for it, like for similar structures in our inventory. But it's not that simple to send compounds if they're not from like a specific project or anything. So unfortunately, it's very difficult. But I can still look for it and try again. But it's been 6 months since I asked for one compound, and I'm still waiting for the legal reply to that. So
that's from the NDI, right?
Yeah, that's from the NDI. It's complicated because we have to send compounds from any of our projects. We have to set up an MTA, a material transfer agreement between the parts, and to do that, the legal has to do like a whole I don't know, checking everything, and it's kind of complicated because of.
Louisa, do you expect the same to happen? Sorry, carry on.
No, it's fine.
Would you expect the same to happen with the SAR gap for phenomenal paper? Those compounds that we collected.
No.
Like 11.
No. If it's done, it's fine. I'm sure that Ben was involved in this, and it's all good. It's just that for sending compounds like solid samples from one center to the other, we need to do this. And for I don't know, I don't know what reason, it's very difficult to do. But there's no problems. If the paper is there, if Ben was already on top of it, that's fine.
No, the SAR gap analysis, the scheme that me and Han created.
I
Those,
think that's
So you you think about like the paying for it?
no,
because
is
you
there
guys
going to
are
be a
buying it,
problem with
no?
getting those compounds for extra testing that we derived from the paper?
Yeah, I think Matt has an agreement with Epigam for that via Ben
Yeah.
setup then.
Yeah,
Okay.
so that's fine. It's just like
I was just reconfirming. Mhm.
so okay. So because I thought that you guys are buying those compounds from Enemy or something like that, or no?
No.
Oh no, it's from Epican. No, that's fine. If it's already done, it's good. Like Epican, it's worked with us for many years, so it's fine.
No, this was also in relation to the fembendazoles. I found a paper where there were many already active fembendazoles, and I asked Louisa for help for that.
Yeah.
There was another question which was also in the previous meeting, but I guess you could still help there, Louisa. Ben calculated the log D for us using a program, and we were wondering if you could still do that for us.
Yeah, absolutely. It actually, that's a yes. Send me the compounds. I'll be happy to help with that. But also there is one thing that I mentioned to Wendy, to Bernard, and to Matt in one of my last emails last week, was about DMPK, in vitro DMPK. So we have this partnership with the CARE, the University of Dundee. So Wellcome Center for Anti-Infective Research. And we have a really good connection with them. And so they often host fellows from all over the world for train then into doing like this in vitro DMPK essays. And they have one fellow coming this July, and they wanted to, usually they use their own proprietary compounds from Dundee to get the training for these fellows. But Lauren Webster, which is a very good friend of the NDI, she suggested that maybe we could use our open science projects, such as the MycetOS or the OSN, to send compounds to Dundee to get in vitro DMPK, either for like students to know what the assays are, what kind of structure property relationship they can draw from that, or for publication wise. So it's up to you. So it's something on the table. So if you want to get in vitro DMPK for free, I think they can offer LogD, maybe some of the intrinsic clearance, maybe RAT or mice. I'm not sure. Maybe permeability as well, and solubility. So this is something that it's on the table. If you want to do that, I can do the bridge between MycetOS and Lauren. She's happy to do any sort of I don't know, introduction, share protocols and everything. We can set up that for sure. It's we still have a couple of months yet, and if the compounds are going from Europe, I'm sure it's pretty fine. It's fast shipping compounds, so that's fine. Just let me know what you guys want, and I can certainly just do the connection, and you guys can do it. So, but I'm happy to calculate log D as well. Star drop.
That sounds great.
Yeah, for me it makes sense. Either if you want to do that with students or to publish, it doesn't matter. I think it can get like a lot of data, and it'll be very good for, I don't know, for everything. So just let me know what you want, and then I can do that.
Thank you, Louisa. Moving on to the next part, because we're a bit short of time. We only got like 9 minutes left. There was also a point about the school projects with where Hank and was supposed to talk to Alice and Peter about this, about the senior honorary students to join the GitHub. Maybe any quick news on this?
Hi, Dimitri. So it's Marita has joined us in the meeting, so I would like to invite Marita to introduce herself and let you know what she's working on. And then if we have time, I'll take a moment to speak to Payan's work, who's not able to be with us this evening, if that's okay.
Sure. Hello, Marita.
Hi, sorry. I have a few slides that I can share, just some intro slides. Yeah, so I just started my honours project this year as part of the Rutledge group at the University of Sydney. And I'm planning on working on the benzimidazoles series. These are just a few ideas that I had. I haven't fully figured out if all of them are viable yet. I'm currently working on the lead compound in the black box. So this is sort of the synthesis I've been working on. I'm still in the beginning stages of figuring out step one and optimizing those reaction conditions. I think Hung has worked on this before, so I'm also picking up some of the stuff that he was working on as well. But yeah, I've just sort of started working on this particular compound in this series, and I'm hoping to see if I can create some variety of this lead compound. Yeah, that's it.
Great. Good luck with your synthesis then. Hope to see some new molecules.
Thanks, Marita. And Payan, who I think will join us in an upcoming meeting. To begin with, she's going to work on the chiral resolution of some of the phenarimal compounds, starting with the most, or hopefully some of the more active ones, so we can better understand if one enantiomer is much more potent than another, which I don't think we have much or any information on to date. And then we can see what the results of that is and whether we want to proceed with any enantioselective synthesis. So I hope that shares a brief overview of what she's going to be looking at.
In terms of the benzimidazoles, I think Wendy was asking on GitHub if there's albendazole available anywhere. Am I right, Wendy?
Yeah, I made a whole list of which I found in a paper, which I found interesting to test because those are already relatively far. They are already tested in vivo for most instances. And I thought it's a low-hanging fruit to test those as well.
Maybe we can follow up on that, Marita, and see whether they might be within scope for you to have to try and tackle as part of your project. So we'll have a look for that, Wendy. And thanks, Bernard, for sharing.
Yeah, Marita, you can also post the slides onto the comment section of the GitHub issue for today's meeting. That will be great in just helping everyone understand where you're up to, and like if you have any question, you can also just drop some there as well. Or, like,
yeah, sure.
I understand, and we'll be happy to help as well.
Yeah. Okay.
I just posted the link to today's meeting, so just in case, in the comment section. We have a few more points to go through. I know we don't have time probably to go through all of them. Walid, do you have any information? Is Walid in today or not?
Walid just became a new parent, so he said that it would be difficult for him to meet today. And for the other remaining points from our side, it's quite easy. We just ordered some libraries, which we will test soon. Mickey is going to test the kinase set. And when we have the data, we will upload it again. Probably we will make for the kinase set a new tab in the spreadsheet.
I see. And that refers to kinase inhibitor library as well.
Yes, and also Rowan's compounds arrived, but not tested yet. And the global health box we already presented.
Okay, just to finalize and wrap up like the whole meeting, I would like to give like a very brief, short update on what's happening here at UCL in terms of the Keroxime Series 6. So as you know, in March, I sent a large batch of compounds to Wendy, so we will be waiting for the potency results. It will be the first batch of Keroximes where we have separated the E and Z isomers. So it will be very interesting to see what the potency results would be like. In the meantime, I'm filling some SAR gaps and doing a bit of methodology for the thesis that I will be writing shortly. Apart from that, I'll just share the screen quickly, just for 1 minute. In terms of something new, there were 2 molecules that have been synthesized before, and which turned out to be not what we thought they were initially. So I'm talking about the DM82/1 molecule, which was which was a double alkylated product, but at nitrogen region, so which gave a pyridinium type like compound, but actually this could not be confirmed by the NMR. And it turns out, and I confirmed this with depth 135 carbon NMR, that the double alkylation happens at the CH2 linker, which is actually giving this compound highlighted in blue. So in principle, this means that we have a totally different Keroxime compound with the same scaffold, but with double alkylation, which would be very interesting to test. But again, we need to be a bit careful with both with log D values, because it might skyrocket depending on which substrate we use. But I would still send them to Wendy and Ma in the next batch, which should come shortly, sometime in the summer. And also this applies to the DM88 compound. So this one was synthesized as usual with with normal single alkylation, monoalkylated product. But then I got a confirmed variant with double alkylation of the CH2 linker, and the pyridinium salt did not prove to be present as such. There was one case where I got a triple alkylation, and I still have it in the crude mixture. But I haven't purified it yet, and I think it's not a good idea because the molecular weight would be very too high. So me and Matt, we think it's not a good idea to purify this one unless anyone has any objections or suggestions. Bernard, what would you think? Again, it's like a bit hard to say, but we think it's a bad idea.
Yeah, it's hard to say. Well, there are some active pyridinium compounds, antiparasitic or antifungal active compounds of that type. Yeah, of course, its molecular size is out of the range of the Lipinski rules, I suppose, with 680. But well, there are also many platinum complexes which violate these rules. So if you want to purify it, why not? Maybe it's active. Let's see.
I think you think of it from like a practical point of view. So the in vitro test can be done quite in a high throughput, as I think Wendy mentioned before. So if it fits with your timeline, and why not just give it a go, unless it's like super difficult to separate it. It doesn't hurt to just send it with your next batch if you can complete.
So one of the major problems with Keroximes is that actually the alkylation reactions did not go as I would expect them to go. So I did a lot of methodology, because simply doing an alkylation reaction under standard conditions gives like a lot of side products. That's the main problem. So those were not separately, just for understanding, separately synthesized. They came as a mixture of those compounds in some cases. And that was the difficulty. So the lower yields, difficult separation, plus if you get like double alkylated product, it's E and Z at the same time. So yeah, it's a bit of like time investment in that sense. Yeah, but just the update for today's meeting is that we have like 2 extra compounds that were like out of the table, but now they confirmed and they will be ready for, and they are already ready for the next batch, and we'll send the double alkylated for sure, the blue ones for sure. Yeah, that's all for UCL update for today's meeting.
Yeah, just on that note as well, I'll just give a brief update from UCIT. So I will be sending a batch of phenarimones, the final batch that I will be making as part of my PhD. So it's about 12 compounds. I will also double check with Clementine, Marita, and Payan if they have any compounds they would like to be shipped with my batch as well. So that will be going to Wendy this week, and hopefully you guys will have that soon at Erasmus. I will also when I get to ship that, I will provide the structure on the comment section as well. So update, as well as I will update the master list with those structures as well. So yeah, just
I will update the master list with my compounds as well, and I would like to ask Louisa to help with the log D value calculations, because that's very important information. It's a bit missing, and usually for catexymes they're much higher. At least the correlation was that those compounds that proved potent in vitro, and for the IC values that we got like very good potency, the log D values were much higher than for phenarimols. So it kind of for catexymes, at least the correlation is not like direct with phenarimols, at least for now. But we need more data on this, so we just need to be a bit more careful on the calculations as well. And I guess that's it.
No, I'll do that, Doris.
Yeah, thank you. So unless anyone has anything else to add, we can finish this meeting today.
Yeah, just one thing to note, as I will be an apology for the 23rd of May as well as the June meeting. So I will be focusing on finishing my thesis. So I will be of limited help during these 2 months, but I'll be back in July if there's anything that still needs my attention. So just a heads up.
Good luck with writing your thesis.
Yeah.
Yeah, good luck, Hank. Yeah. On the 23rd of May, I'll try to, by this time, to have a finalized with Matt whether we are going for Southern Force for the project, because it might change how the final months of my lab work would be. So we have to decide this rapidly as soon as possible, and we will do this and update you as SAP on this decision. Okay, then thank you very much for attending today's meeting, and I will see everyone on 23rd of May.
Thank you, Dimitri. Bye-bye, everyone.
Thank you. Goodbye.
Bye.
Thank you.